A group of scientists at the University of Connecticut set out to cure multiple sclerosis. They gave mice a drug combination, a popular one that thousands of people are already taking right now voluntarily because they read it extends lifespan by 36% and clears out the cells that make you age faster. The scientists were trying to help. What they found instead was that the drug had eaten the mice's brains, not the neurons, the insulation around them, and the younger the mouse, the worse the damage. The combination is called D plus Q, dasatinib and quercetin. Two drugs taken together on a specific schedule, a few days on, a few weeks off, repeat. Dasatinib is a cancer drug, originally developed for leukemia. Quercetin is a flavonoid. It's in onions, apples, red wine. You can buy it at any pharmacy right now. Neither of these facts fully prepares you for what they do together, or why serious researchers got excited about them. The idea behind D plus Q is called senolytic therapy, and the theory it's built on is one of the more compelling ideas in aging research right now. So, the first thing to understand is why anyone is taking this in the first place. As you age, your body accumulates what researchers call senescent cells, zombie cells, cells that should have died, cells that stopped dividing, stopped functioning, but refuse to go. They just sit there, and while they sit there, they secrete a constant low-level stream of inflammatory chemicals into the surrounding tissue. This chronic inflammation is one of the current leading theories for why aging does what it does, why your joints start going, why your kidneys lose function, why your brain starts to slow. The zombie cells are not directly killing you, they are just making your entire body slightly worse, all the time for decades. D plus Q's job is to kill them. Dasatinib blocks one set of survival signals the zombie cells use to stay alive. Quercetin blocks another. Together, they are more effective at clearing zombie cells than either drug is alone. The research behind this is not fringe. In naturally aged mice, D plus Q extended post-treatment lifespan by 36%. It reversed symptoms of frailty, kidney dysfunction, pulmonary fibrosis. Multiple clinical trials are active right now in Alzheimer's disease, MS, chronic kidney disease, and others. Longevity researchers treat D plus Q as one of the most evidence-backed tools available. Brian Johnson has made cellular senescence a central pillar of his protocols. The Peter Attia adjacent world discusses D plus Q openly. This is not pseudoscience. This is the frontier. The second thing to understand is what these drugs actually are, because this matters later. Dasatinib works by blocking proteins called kinases, molecular switches that tell cells what to do. It was designed to block the switches that tell cancer cells to keep growing and dividing. At full chemotherapy doses, taken daily, it has a known side effect list that includes nerve damage, vision problems, memory issues, and confusion. At the much lower senolytic dose, taken intermittently, a few days at a time, the assumption was that the side effect profile would be far more manageable. The hit and run approach. Go in, kill the zombie cells, come out. Let the body recover. Quercetin is the one that feels harmless. It's a supplement. It's in your food. The quercetin supplement market is worth nearly $2 billion. People buy it for immune support. They don't think of it as a pharmacological agent. Together, at the standard research dose, 100 milligrams of dasatinib, 1,000 milligrams of quercetin, thousands of people are currently self-administering this combination. Some from longevity clinics, some ordering dasatinib through international pharmacies, some combining a prescription they got for unrelated reasons with a supplement they picked up at the health food store. Nobody told them to stop. The research said it was working. And then Stephen Crocker's team published their paper. The third thing, and this is the one that matters, is what actually happened in the study. Crocker's lab was not trying to prove D plus Q was dangerous. They were testing whether it could help MS patients. The hypothesis was optimistic. Maybe D plus Q could clear the zombie cells that were blocking myelin repair in MS and help patients regenerate the insulation their nerves had lost. To understand why this went wrong, you need to know one thing about the brain. Your neurons, the nerve cells, don't run naked. They're wrapped in a fatty insulating layer called myelin. Myelin is what makes signals travel fast. Without it, signals slow down, blur together, and lose strength. Multiple sclerosis is defined by myelin loss. When the myelin goes, coordination goes, cognition goes, the signal chain starts to break. Myelin is made by specialized cells called oligodendrocytes. Their entire job is to extend branches out to nearby neurons and wrap them. Think of them as the insulation crew. They are constantly working, producing myelin, maintaining it, extending coverage to neurons that need it. Crocker's team gave mice D plus Q at the standard research dose. Young mice, middle-aged mice, aged mice. Then they looked at the brains. The corpus callosum, the thick bundle of fibers connecting the brain's two halves, the central communication highway, had visibly deteriorated. Under the microscope, healthy neurons showed dark rings around them. That's the myelin sheath. In the treated mice, those rings were dramatically reduced or gone. The insulation had disappeared. Every mouse, both age groups, standard dose. This is the part that gets strange. The researchers expected to find dead oligodendrocytes. When cells are damaged, they die. That's how most drug toxicity works. The oligodendrocytes weren't dead. They had pulled back, retracted their branches, reduced the area they were covering, and, at the molecular level, shifted back to behaving like immature juvenile cells. Cells that haven't yet developed the full capability to produce myelin. Crocker described it as the cells reverting to a younger state, but less functional. The drugs were choking off the energy supply the cells needed to do their job, and the cells responded by doing less job. And then the finding that changed the shape of the whole story came in from the younger mice. They were expecting aged animals to show the worst damage. That's how almost every drug toxicity study goes. Older biology, more fragile. The data said something else entirely. The fourth thing, and the gut punch, is this. The younger the mouse, the worse the myelin damage. This runs against every intuition. Young, healthy animals should be more resilient, more capacity to repair, more robust baseline. That's how it's supposed to work. The reason it went the other way is actually the explanation for everything. Young oligodendrocytes are working harder. They're actively building myelin, extending branches, producing the proteins that make insulation. More metabolically active, more output. When D plus Q cuts off their energy supply, the cells that are doing the most work have the furthest to fall. The aged cells were already in a lower output state. They had less activity to lose. What this means for real-world use, the demographic most enthusiastic about preventative longevity protocols, people in their 20s, 30s, and 40s taking D plus Q to get ahead of aging before it starts, are the demographic the mouse data suggests would be hit hardest. And here is the part nobody can answer right now. No one has looked. No published study has examined corpus callosum integrity or oligodendrocyte health in humans who have taken D plus Q. The clinical trials that exist were not designed to detect this. The phase 1 Alzheimer's trial showed no significant changes in any Alzheimer's biomarker. A small 2024 study found D plus Q actually increased biological age on certain measures and reduced telomere length, the opposite of what a longevity drug should do. None of these studies were looking at myelin. Nobody knows what D plus Q does to the human corpus callosum because nobody has ever looked. They were too busy studying whether it worked. The researchers who ran this study were not trying to find a problem, they were trying to help people with MS. They gave mice the exact dose used in longevity research, not an overdose, not an experimental extreme, the standard protocol, and produced brain damage that looked structurally identical to MS lesions in every animal they tested. The most disturbing part of the Crocker paper is not the finding itself. The most disturbing part is how it was found. Accidentally, by a team looking for something else. D plus Q did not have a secret. It had a gap. A specific measurement nobody had thought to take, in a tissue nobody had thought to check, in a population, healthy, young, preventive use humans, that most of the trials weren't designed around. Thousands of people are currently taking this combination. Most of them found it through the same research that made it famous: the 36% lifespan extension, the clinical trials, the longevity influencers. The evidence is real. The theory is sound. The gap is also real. The honest answer is that the gap exists, and nobody is monitoring for it. And the study that revealed it was published in March 2026. The researchers who found this were trying to fix multiple sclerosis. Instead, they accidentally built the most accurate laboratory model of it anyone has ever produced. What that means for the people already taking this, nobody knows yet. That part is a different study.